Use this protocol to make neuronal progenitor cells from patient skin fibroblasts and study neurological and neurodegenerative diseases. We feel like this protocol has a lot of advantages over classic iPS technology, including the speed, but also getting in certain cases a more severe disease phenotype. We study neurological and neurodegenerative diseases, and using skin samples allows us really to evaluate the diversity of the patients as well. My lab is particularly interested in the role of astrocytes in diseases, and we use the astrocytes that we make from the NPCs to test medications and also to study disease mechanisms, but also in co-cultures with human and mouse neurons. We hope this protocol is going to be informative, and please reach out if you have any questions. Thank you.
Use primary human skin fibroblasts that can be obtained from cell banks or skin biopsies. Culture cells at 37 degrees Celsius in a 5% CO2 tissue culture incubator. Passage cells for at least 1 to 2 passages post-thawing prior to use in the experiment. Once the cells are ready, coat a well of a 6-well plate with human fibronectin at a 1 in 200 concentration, diluted in PBS at room temperature for 15 to 20 minutes. Prepare fibroblast media as shown in Table 1. When the cells are ready to be plated, remove the fibronectin from the dish and immediately add cell solution, or 2 ml of fibroblast media. Do not let the dish dry out prior to adding media. Depending on how fast the cells grow, plate 150,000 to 200,000